Hello everyone, my name is Carissa Slaughter. I'm a biology major and my CERT 2021 project is over Trichomonas vaginalis induced toll-like receptor gene expression and cervical epithelial cells. And Dr. Ashwini Kucknoor is my mentor. So our hypothesis for this project is to analyze the TLR or toll-like receptor gene expression in the cervical epithelial cells induced by Trichomonas vaginalis. So we know that Trichomonas, it elicits host inflammatory responses that promote cell proliferation or makes many copies of the cells and dysregulation of the cell's cycle. So we know that Trichomonas has correlation with cervical epithelial cancer. So our goals for this project were to better understand the cytokine expression, then to analyze the toll-like receptor activation, the molecular mechanism, and to study the innate immune responses that are induced by Trichomonas vaginalis, and also to potentially provide targets against Trichomonas infections for chemotherapy. So to begin, you can see that Trichomonas vaginalis it swims around, so it's a protozoan, it's a parasite, it is extracellular, so it's outside of the cell, it is lophotrichously flagellated, meaning it's on either side, it's got flagella, it's unicellular, so it's one cell, sexually transmitted disease, and it affects 180,000 people globally. And you can see that it disturbs the vaginal microbiota, where it's located right here. So next I'm going to introduce you to the Henrietta Lacks cells or HeLa cells, and these are cervical cancer, and you can tell that in this picture that I took with the light microscope that this is completely packed. It's a monolayer of the tissue. So it's essentially it is in the flask it is there's a treated side, and the HeLa are directly connected to that treated side. And that's where they grow. So toll-like receptors are super important for this, and basically toll-like receptors are the innate immune system receptors that identify the pathogens like Trichomonas to determine the molecular mechanism for the host defense. So for what we would give as a host defense. So we are looking at this top layer, the extracellular, that's what we're focused on. So where it says like 1, 2, TLR26, 2, 4, 5. So we're going to be looking at the TLR4, and that is a lipopolysaccharide TLR. And the reason why is because we've seen that there are correlations with other protozoan pathogens that have a good correlation with TLR4. So we thought that because Trichomonas vaginalis is also a pathogenic protozoan, that we would also take that extracellular toll-like receptor. So next we're going to be looking at the culturing of Trichomonas. So basically you have Diamond's medium, and you're putting horse donor serum along with that. And you can tell that in the left video I'm making the different media, and then I'm culturing it in the two vents next to it, and the incubator is what it looks like here in the middle. And then you have HeLa cell culturing, which is pretty similar, and the main difference is that it's a DMEM, so it's like a red media compared to the yellow, a Diamond's media. And I'm also using fetal bovine serum instead of the horse serum that I use in Trichomonas. These are a little bit trickier to culture and get to grow. Alright, so next we're going to be talking about the counting of the cells. So we use this tool called the hemocytometer, which is a 5x5 where we count the cells. And you can tell to the left in this video I'm doing lots of math after I'm counting. And we do a 1-to-3 ratio. So 1 being the Trichomonas, 3 being the HeLa. And basically we do a lot of math and preparation for the interaction. So next we're going to be talking about the interactions. So with the interactions, over here to the left, you can see the Trichomonas. It's very fast, but it is swimming. I'm going to replay it a few times. So it's a fast video of the Trichomonas swimming, if you want to call it that, on top of the HeLa. And the rest of these are just the interaction. I have a manual pipette, that I am injecting a 1-to-3 ratio of the Trichomonas on top of that treated side where the HeLa has already grown. And we did that for about 30 minutes as an interaction. All right, so then after that, you collect and you do the trypsinization. So trypsin is an enzyme, and basically you take that treated side and that trypsin is going to scrunch up the HeLa and take it off of that edge, and then we're going to do the centrifugation, which spins it very, very fast to where it separates the pellet at the bottom of the HeLa, and then it has the rest is going to be a supernatant or conditioned media. So I'm going to take the conditioned media into one, and then I'm going to have the trypsinized centrifugation HeLa pellet. So that's what we do. And next we're going to talk about the RNA isolation. So from that pellet, we go into the RNA isolation to prepare it for the RT-PCR and the PCR, which I'm going to talk about in just a minute. So after the RNA isolation, we do those different tests and we run it on a gel. It's called gel electrophoresis. And basically, that determines it separates the molecules based on the size. So we're able to determine the different sizes. All right, so after the gel electrophoresis, all right, so we do have a lot of immunological techniques that I've showed you, and we have 4 main experiments. So one is a polymerase chain reaction, then we had the reverse transcription polymerase chain reaction, then the ELISA or the enzyme-linked immunosorbent assay, and then we had the adherence assay tunnel staining. So for the polymerase chain reaction, experiment 1, we use this right here on the left. This is a polymerase chain reaction machine. It basically is the copy machine, is how I say it, and it isolates the genomic DNA from HeLa. That's what we do first, and it amplifies the genes using the primers that we synthesize, and we ensure that it's in the right conditions. So we determine all the annealing temperatures. So you saw all the intracellular and extracellular TLRs that was in the beginning, and we use those primers and we determined the correct annealing temperatures with all of those. And then we have the RT-PCR, the reverse transcription polymerase chain reaction. So it's in the same machine that we use for the PCR, but it has a different role. We're looking for the gene expression. That's our analysis, and we isolate that RNA from both the HeLa and the interacted Trichomonas 6 and Trichomonas 1, 2, 3 plus the HeLa in that little pellet after the centrifugation. And if it has enough messenger RNA, the reverse transcription does occur, and the brighter bands that you see on here, they indicate more amplification and more expression. So TLR, we could conclude that at higher rates, it's found in the TV123 than, and I'm going to show you here in this picture. There's 2 different arrows that I'm popping up, and that is the TLR induced at higher rates in TV123 than the TV plus HeLa and the HeLa alone. And this gap over here is our internal control, and that TLR4 is obviously the primer of the toll-like receptor. All right, so looking at the ELISA, which is that enzyme-linked immunosorbent assay, we are looking for detecting pro-inflammatory cytokine, interleukin 6. So whether this research is valid, we're looking at it from that conditioned media or supernatant for the interleukin 6. So this is, the induction of IL-6, and we found from this that TV6 plus HeLa in that conditioned media and the HeLa alone, this is in the bottom 2 right graphs right here, that they showed the basal level, so the very bottom level, while TV123 plus HeLa, that interaction, showed an increase in the interleukin 6 pro-inflammatory cytokine, which causes vaginitis because there's inflammation within the vaginal microbiota. So we're seeing pretty consistent results, higher rates for both. So because of that, this is, the conclusion. We know that Trichomonas vaginalis, it induces toll-like receptor 4, innate immune receptor, and also the interleukin 6 pro-inflammatory cytokine. We found that in the experiment 1, 2, and 3. So our future direction going forward, we're going to do an adherence assay, and we expect a higher rate for TV123 adherence because of the increased expression and the toll-like receptor, which we discovered in the reverse transcription polymerase chain reaction, and the increased expression of the interleukin-6 and the ELISA using fluorescent markers. And then we also are going to look at the tunnel staining to ensure that HeLa is not dying or having apoptosis. And I want to thank the Office of Undergraduate Research at Lamar University, and for sure Dr. Kucknoor. She has been incredible throughout this whole experience. I'm very thankful for her. Anytime I needed anything, I could give her a quick text, and she was very quick to respond if I needed a call. So I'm very thankful for the role that she's played in my career so far. And also for Dr. Brim. He's been very great. And I'm very thankful that he has also been very responsive and helped with any type of things that we needed. So thank you again to the Office of Undergraduate Research at Lamar University. This Summer Undergraduate Research Fellowship 2021 has been an incredible experience. Thank you. Bye-bye.